Psuedogenes. An inactive but stable component of a genome resembling a gene, apparently derived from active genes by mutation. Transcription factors. A protein that regulates the transcription of genes. Alternate splicing. Phenomenon of splicing an RNA transcript in different way as a ways making it possible for a single gene to encode different polypeptides, A, X, intron, exon, intron, ... The splice can occur at end of both introns, removing the exon as well. B, exons equals coding sequences, introns equals non-coding sequences. Inducer. A substance of low molecular weight that is bound by a repressor to produce a complex that can no longer bind to the operator. Thus, the presence of the inducer turns on the expression of the gene controlled by the operator. Heat shock proteins. A group of proteins that help to stabilize the internal cellular environment, B, found in both prokaryotes and eukaryotes and are among the most conserved polypeptides known, C. These proteins are synthesized when organisms are subjected to the stress of high temperatures. D. Expression of heat shock proteins is regulated on transcriptional level. Heat shock transcription factor. Present in the nuclei of Drosophila cells. When the Drosophila are heat-stressed, the HSTF is chemically altered by phosphorylation. This altered state then binds upstream of the HSP70, making the gene more accessible to RNA polymerase II. HSTF binds to HSEs. Histone Acetyl transferases. Transfers an acetyl group to the lysine in histones. Kinases transfer phosphate. SWI/SNF regulates transcription by moving histone octamers along the DNA molecule. HDAC remove acetyl group from lysines. MT transfer methyl groups to lysine, arginine, and histidine in histones. Heat shock response element. The sequences on RNA that HSTF binds to make the gene more accessible to RNA polymerase I. Hormone. An organic product of cells of one part of the body that is transported by the body fluids to another part where it influences activity or serves as a coordinating agent, circulates through the steroid hormone. Small, lipid-soluble molecules derived from cholesterol. Lipid natures allows them to pass through cell membranes easily. Inside the cells, steroid hormones interact with cytoplasmic or nuclear Proteins called hormone receptors. Receptor hormone complex then interacts with the DNA and acts as a transcription factor to regulate the expression of certain genes. A. X. Estrogen, progesterone, testosterone. Hormone receptor. Cytoplasmic nuclear protein that interacts with the hormones. Once complexed with a hormone, the complex acts as a transcription factor for DNA and regulates the expression of certain genes. Peptide hormone. Linear chains of amino acids encoded by genes, too large to pass freely through cell membrane, so they must interact with membrane-bound receptor proteins. Interactions causes a conformational change in the receptor which initiates a cascade of changes in other proteins from the cytoplasm to the nucleus, ultimately resulting in regulating the expression of specific genes. Membrane-bound receptor protein. Receptors that interact with peptide hormones. Signal transduction. The process whereby a molecular signal such as a hormone is passed internally within a cell by a system of molecules to effect a change in the cell's state. Hormone response elements. Analogous to heat shock response elements, specific sequences on DNA that respond to hormone-induced gene expression. Enhancers. A DNA sequence that influences transcription of a nearby gene. Zinc Finger. A short peptide loop when two cysteines from one part of the polypeptide and two histidines in another part bind to zinc ion, one of the transcription factor motifs. Helix-turn-helix. Stretch of three helices separated by turns, one of the transcription factor motifs. Leucine zipper. Dimers between two polypeptides that each have leucine at very 7th position. Base helix loop helix. A stretch of two helical regions and basic region. Homeodomain. An amino acid sequence in the polypeptide to which the homeobox corresponds to. The homeobox is a DNA sequence found in several genes that are involved in the specification of organs in different parts and animals. Chromomeres. Small bodies that are identified by their characteristic size and linear arrangement along a chromosome. Ecdysone. A hormone that influences development in insects. DNase I hypersensitivity sites. specific sites on isolate chromatin that are cleaved by low concentrations of DNase I actively. Transcribed DNA is more open to nuclease attack. These sites may mark regions in which the DNA is locally unwound or where transcription begins. Many are located on the LCR. Locus control region. 15 kilobits long, location of DNase I hypersensitivity regions, contains binding sites for transcription. Factors that pre-activate the individual gene for transcription, orientation-dependent functionality, but can work on different chromosomes. Chromatin remodeling. The alteration of the structure of DNA and its associated protein molecules, especially histones, by a protein complex. This remodeling often involves the chemical modification of the histones. Heterochromatin. Chromatin staining darkly even during interphase, often containing repetitive DNA with few genes. Repressed gene function, because its condensed form is not accessible to the transcriptional. Euchromatin. Genetic material that is not stained so intensely by certain dyes during interphase and that comprises many different kinds of genes. Position effect variegation. Phenotypic variation within an individual that is due to a change in the genomic position of a gene. Usually this type of variation is seen when a gene naturally located in euchromatin is moved by a chromosome rearrangement to a heterochromatin region of the genome. Silencers. A DNA sequence that helps to reduce or shut off the expression of a nearby gene. CpG islands. Clusters of cytosines and guanines that often occur upstream of human genes, situated near transcription start sites. Rarely methylated equals conducive to transcription. DNA in the vicinity of a CpG island is hypersensitive to digestion with DNase I. Imprinted. A process that alters the states of a gene without altering its nucleotide sequence, often associated with methylation of specific nucleotides in the gene. The altered state is established in the germline and is transmitted to the offspring, where it may persist throughout the offspring's life. A gene that has been altered in this way is said to have been imprinted. Gene amplification. A phenomenon whereby the DNA of a specific gene or set of genes is replicated independently of the rest of the genome to increase the number of gene copies. Exon activation center. the location at which X chromosome inactivation begins and then spreads in opposite directions toward the ends of the chromosome. 11th gene. An inactive X chromosome transcribes RNA that then coats the chromosome. 11th equals X inactive specific transcript. mRNA. Double-stranded RNA molecules 21 to 28 base pairs long that mediate the phenomenon of RNA interference, also known as micromolecules. 37. RNA interference. A phenomenon in which double-stranded RNA prevents the expression of a gene homologous to at least part of the RNA. Inactivation. Inactivation of X chromosome in XX female mammals is mediated by a non-coding RNA transcribed from the 11th gene on that chromosome. Hyperactivation. Hyperactivation of the single X chromosome in male Drosophila is mediated by an RNA protein complex that binds to many sites on that chromosome and stimulates the transcription of its gene. Hypoactivation. Hypoactivation of the 2X chromosome in C. elegans hermaphrodites is mediated by proteins that bind to these chromosomes and reduce the transcription of their genes.